This is the first video of the Cell Death Pathways lecture. The objectives of this lecture are to understand and discuss different cell death pathways and to link key molecules to specific cell death pathways that are relevant for neurodegeneration. This is the outline of the lecture in which we will discuss general notions of cell death or neuronal death and the different types of neuronal death. Those include necrosis, apoptosis, necroptosis, parthanatos, and autophagic cell death. For this video, we will discuss the general notions of cell death and talk in more detail about one of the first types of neuronal cell death, which is necrosis. So neuronal death is normal during nervous system development. It's a needed and necessary process to ensure the proper development of our nervous system. However, it can be abnormal in brain and spinal cord disease and injury. So a better understanding of the molecular mechanisms of neuronal cell death in the nervous system during injury and disease can lead to new therapeutic approaches for the prevention of neurodegeneration and also neurological disabilities, and will expand the field of cell death biology. In this slide, we can find very basic characteristics of neurodegeneration. Those include metabolic imbalance, processes retraction, synaptic loss, synaptic dysfunction, protein mislocalization and aggregation, and neuronal death. So understanding all these processes and how they lead to neuronal death are key to ensure that we understand what is happening in neurodegeneration and how can we intervene. There are different conditions that can cause neuronal death, and here there is a very basic list of some of those factors. One of the conditions that causes neuronal death are insufficient trophic factors, and that is particularly relevant during aging. Another example are direct injury, such as TBI or ischemia, and those also cause neuronal death. There are other factors such as dysregulation of the internal milieu, such as elevated calcium, DNA damage, accumulation of reactive oxygen species, and all of those factors can cause neuronal death. And then the last factors that we have in this list are genetic mutations that cause toxic gain or loss of function, and also can cause protein misfolding and aggregation. And in those, we can find many of the neurodegenerative diseases that we know, such as Alzheimer's, Parkinson's disease, Huntington's disease, ALS, and many others. So now we will start talking about the different types of neuronal cell death pathways. And we will focus on necrosis. So necrosis is considered a deleterious phenomena that can actually be blocked by suppressing the stimulus. So it is a process that can be stopped and it can be interrupted once you identify what is causing the necrosis initiation. This is a form of a traumatic cell death that results from an acute cellular injury or an internal exposure of high concentrations of oxidants, ionophores, or different types of lesions. During very long time, necrosis has been considered as an unprogrammed cell death. However, now we know that there are different forms of programmed necrosis, especially those that are related to glutamate excitotoxicity. And in the next slides, we will talk in more detail about this specific programmed necrosis. Another characteristic of necrosis is cellular swelling. And in this picture, you can see the morphology of a necrotic tissue or a necrotic cell, which is mainly due to cell lysis following the rupture of the plasma membrane. Here, there are some other basic characteristics of necrosis. Um, Necrosis, it's always a pathological pathway, and it affects not only the cells that initiate the necrotic pathway, but also adjacent groups of cells. The cell size is usually increased during the necrosis process due to cell swelling, but it's considered a passive process because it's not caused by de novo protein synthesis. However, there are some traumatic processes that include histone and DNA degradation and plasma membrane disruption. And all of this ends up in inflammatory reactions or inflammatory processes. So now let's talk about this very specific programmed necrotic pathway. So as you know, glutamate is the major excitatory neurotransmitter of the central nervous system. And we've seen in previous classes how glutamate interacts and activates different types of receptors, ionotropic receptors such as NMDA receptors or AMPA receptors, and also metabotropic receptors. And under normal conditions, glutamate that is released from presynaptic terminals reacts with this or interacts with these receptors and causes calcium influx, which promotes neuronal depolarization. However, glutamate can cause severe damage, and this process is known as excitotoxicity. Excitotoxicity involves the excessive release of glutamate from presynaptic nerve terminals and also from the reversal of astrocytic glutamate uptake. So this causes an excessive neuronal depolarization. NMDA receptors are activated in postsynaptic neurons as well as other receptors, opening their receptor-operated cation channels and allow a massive calcium influx and also calcium accumulation inside the neurons. The calcium influx activates different enzymes like proteases, such as scalpain, and the neuronal nitric oxide synthase, or NOS. And these processes induce osmotic swelling, but also activation of mitochondrial disruption. All of this increases reactive oxygen species and reactive nitric or nitrogen species, and also stop the ATP production. And these free radicals also damage cellular membranes and intracellular proteins and DNA. And the result of these processes is neuronal necrosis, which is a programmed cell death that is the basis of all acute neuronal injury in the adult brain. So how do we measure necrosis? And what are the different necrotic biomarkers that we can use in the lab to assess whether we're in front of a necrotic cell death pathway? Well, there are different ways of measuring necrosis. Usually, many of them are used in a combinatorial manner. So there probably isn't one particular protein you can use to assess necrosis or one particular test, but rather a combination of different forms or different biomarkers. For instance, a common biomarker for necrosis is the intracellular DNA that becomes accessible after disruption of the cell membrane. So to measure DNA release or intracellular DNA release, we can use propylium iodide, which is a fluorescent DNA binding dye. And actually, this is a routinely used in flow cytometry as a secondary marker for necrosis. Then there are other types of biomarkers that you can test. For instance, calpain 1, the protease calpain 1, is used to determine necrosis and the activation of this protease, which normally exists as a non-activated enzyme, and then during necrosis, it becomes activated through specific cleavage, and then you can detect those products of calpain 1. However, you have to be careful with when using calpain exclusively as a necrotic biomarker because it can also be activated during apoptosis. Then cathepsin B protease activation is also a necrotic biomarker. There are few others that are less known, but right now are getting more attention. Those are not particularly tested in brain necrotic pathways, but in other tissues such as liver reperfusion and ischemia. Those include the microRNA 122 activation, full-length cytokeratin 18 or FK18, and the high mobility group box HMGB1 protein. And then it's also commonly seen the use of the tunnel assay, which is a method for detecting DNA fragmentation by labeling the 3' hydroxy in the double-strand DNA breaks. So this tunnel assay it's also used to determine necrosis, but again, it can also be used as an apoptotic biomarker. So if you really have to determine whether you are in front of a necrotic pathway, you could probably use a combination of propydium iodide staining with calpain 1 expression or activation and HMGB1 protein in the induction, so, like a way of combining different strategies. And finally, I leave you here with a short quiz to revise the most important concepts about this video. We will see this in class and we will talk about any other specific or important concepts that have been revised in this video.